Hello, Tharun.
Hi.
Hi, how are you? And this is Ujjwal. Hi.
Hello.
Hi.
Hi, sir.
How are you?
Fine, sir. How are you?
All right. Is Esmet meeting today? Or is he going to be able to come?
Sir Smith will not be able to join us today. As I told you earlier, we have a conflicting schedule.
Yeah.
So his meeting has starts from 6:30 and mine is going to start from 7.
Okay.
So that's why I was able to join for half an hour.
Okay, that sounds good. So why don't we get into it, and why don't you show me your uh, progress?
Yeah, so I'll just brief over what I done last week. So I trained on a video which had around 50 frames. So the objects had like proper rectangular shapes. So it was easy for the model to like convert into our required what do you call it, images. So it did perform well, and I actually trained for the video in which it had overlapping uh, boundaries or overlappings, but that collab got disconnected, so I couldn't save the weights. So I have to just train it again.
All right. That's good. And Ujjwal, what did you have to report?
I guess the progress that Tharun is making is quite good. So once he was able to do the overlapping label that as we have discussed in the Slack group, I guess next step we are going to me and Ashmit are looking into literature and we saw some steps how to find the corner edges of each and every cell in the model. So there are simple techniques and deep learning model as well. So we have to see which we are going to implement. Also, the last sample that Tharun has sent to us, it has little bit inaccuracies in the form the in the real B there are less number of cells, and in the fake B there are more number of cells in some frames. So it is creating more number of rectangles than the original one in very few cases. So let's see how many frames such frames we got after analyzing the data. We can either skip them because we have to get overall volumetric data basically over the 10 seconds 5 seconds to get the moment velocity and all those things, so we can skip few frames. So once we are done with that, then we can go after scrutinizing the all data, I guess we can go for the rectangle identification, and once we have the coordinates we can start using the basic physics to get all those parameters.
Right.
Yeah, I guess those extra ones actually, it's my fault. Like I actually didn't label them. Those were actually like very blurred or just you couldn't like discriminate. Like two or three would just get combined, so there was no boundary. So it actually did recognize them, but it couldn't recognize them individually. So it has just drawn a single like the whole branch. It has considered a single object. So I guess the data set has a flaw. So yeah,
Oh, so you are saying you are not able to actually label them, but
yeah,
still the
but
model recognizing.
yeah. It could recognize. There was
So
like
it is.
not. It was not a mistake like from the model side. Actually, I did label.
Uh, yeah, so it is basically models performing extremely well. They are also identifying something that is not being able. We are not able to
Yeah,
label them
the
correctly.
edges and all. It can identify it very well because there's like the boundaries and there's no like the background also is like clearly like discriminate. Like you can discriminate it from the objects, so it can
Hmm.
perform very well.
I think
Yeah.
that's very cool. If it is doing that, it will give us more precise data. So yeah, I think we are on right track as of now.
Yeah, once the I train on even the overlapping ones, then we can get a like the final picture, like how where it is working properly or where it is lacking.
Yeah. So what have you done? You've just done like a couple of different. You've taken a couple of different movies and sampled them, taken them frame by frame, and used that as the input data.
Yeah, like so for training I used only one video, and for the testing I used the other video. So that the videos like the objects were like repeating. So I thought that it could like be overfitting or something. So, but it did perform pretty good on the other video, which it did not train on. And like our data also is like very small. I just labeled around 50 frames. So I guess if we increase the data set and include any like a noise or something, then it might get better.
Like the kind of result that we are getting right now with such a few number of frames, me and Ashwin were discussing, we can also pitch it as the like superiority of our method. Like you don't need a whole bunch of thousands of images. You can just take 50 or 60 images and get a very good model out of it. So we can pitch that as well.
Okay. And you're using the technique we discussed like 2 weeks ago. The
Yeah, pix to pix generation.
pix to pix, yeah.
I don't think so anyone has used like this thing in this. Basically, what we are doing is completely opposite of what pix to pix is meant to do. So we are kind of reversing that model, and I don't think so. Like it has been used in such a way before.
Huh.
Because when we started using it in our DL project as well, our professor also gave us same feedback like what you are doing is basically reversing the model, entire model. Uh, you are basically feeding the output and trying to generate the input of that model. So this was kind of a new thing.
Yeah, that's interesting. I know we talked about how people have used it previously. Yeah, it does seem like it's kind of inverted from that. So it'd be interesting to see how it works. Well, it sounds like it's doing pretty well so far. So
Yes, like initial results are better than we expected. So I guess it is going to be fine.
oh, good.
It will work.
Yeah, good. So I
Yeah,
oh, go ahead.
yeah, that's like the PyTorch based model is like better than what they have published. So the paper results which just show we are getting like better results because we are using the PyTorch model. Like they've told that it should give better results. So I guess that is also the reason.
Yeah.
Like you are saying, like they have improved pix to pix. Like obviously there are a lot of improvement in pix to pix as well. Pix to pix is something which was given like around 2015 or 16, and I guess many further improved implementation has been developed.
Ah, yeah.
Good. Yeah. So yeah, I know. So let's go through these data that we have for the Basel area. And I'm almost wondering if it's working that well, if we should try another type of data or another set of images from a different organism to see what those look like as well. I can find some images and to see how it works, like kind of test how robust the model is to like identifying different cell types. We have the C. elegans BIM data, which is like C. elegans embryo, so the cells are pretty easy to make out on those. We have other types of data as well, so I'm going to look around and see if I can find something, and maybe we can run that as well through that once you have that worked out to see if it does good or if it does better or what. I think that would be a good comparison too, to have like two different types of cell. I think we do a lot of stuff with like one cell type or one organism, and I want to sort of push the algorithms a little bit to see what they can do for different types of data.
Yeah, we can check whether the model is looking for these rectangular shapes or it is like just finding out these edges and just creating boundaries around them. So if it's doing so, then it can recognize all the shapes. So yeah, we can test those also.
I think with CD.elegans, so the main problem which we are going to face is labeling them because it is not rectangular anymore. We have to label them in the circular, like oval type shapes. So this is one of the problems. We have to find an appropriate tool for that. If we are able to do that kind of labeling, I think this model will work as well on CD.elegans as well.
Yeah, the labeling part actually what I'm doing is I'm just drawing the polygons around the objects and I'm just getting the JSON files. So this the JSON files has these boundary points. Then I'm just using the pillow library to like plot, create a polygon using those points. So I guess any shape we can label it. It will be it'll be a bit difficult than doing with rectangle. Yeah, so yeah, we can try. But
Yeah, you cannot do like from the C.Elegans, you cannot do it on rectangle. You have to use some higher dimension polygons. Or if I recall correctly, I will share it on Slack as well. I saw a tool couple of days back where you can basically like in the foot like you have if you have used Photoshop, you can basically cut out the images which you have to label by hovering your mouse over the edges of all that. So if I can find that out, I will definitely share it on Slack.
yeah, like in label me, what it does it you can like draw polygons of any number of points. So we just
Oh yeah,
yeah, so
okay.
just click on the boundaries. So then we get those points. Then we can just process those points into a polygon.
Yeah,
You
yeah.
can try. Yeah.
Then I think it will be fine.
Yeah.
Now, Tharun, you said that we had discussed labeling in the Slack, and you had questions. But did you what kind of scheme did you use for labeling?
Yeah, so what I was trying is like when they were overlapping or like one over the other. So what I thought was like they were like the two overlapping, or you can just consider the two as one itself, or something like that. So I think I did overlapping itself. So two rectangles, however they were in the images, I just labeled them as it is.
All right, yeah. Because I remember we had talked about like having like a label where you would say this is the first cell, second cell, and use some sort of scheme like that. But it sounds like you're just using like a simpler technique where you're
Yeah.
just
So
go ahead.
yeah, so I consider all of them as the same object. Yeah, so we can label like we can give like different labels to different yeah objects. But I like thought it was like just simply to just test out the model. I just created a small data set. So yeah, we can do that also.
All right, yeah, that sounds great. Yeah, so that's good. Did you have anything to show, like notebook or anything, or is that pretty rough still?
Yeah, the same. I just share the notebook link, the pix to pix, which the GitHub link that sent. I just use the same thing. I had to just it was like 4 5 lines of code I have to just change to just run the files. It was yeah. I'll share those files as well.
All right. Is there any way you could share your screen on that, or?
Though I don't have it in order. Like I put it in order.
Oh, okay, that's fine.
It was
Yeah.
like 4 to 5 commands. That's it. Just to have to run it in the uh, GitHub. Sorry, the collab. What is it called? The cells. So
Right.
like I just clone the repository and use the same code. So there's not much difference from the original code which they're using.
All right. That's good. All right. That sounds good. That sounds great. Looking forward to seeing more progress on it. You've made some pretty good progress so far. And I'll take a look at we'll keep updating on this, and I can get you some data for another species. You can put that you can see how the algorithm behaves on that, and then we can, I think we can move forward from there. It sounds like we can do some pretty promising, have a pretty promising start here, so we can, I think, move this to something really good. I know there was a little bit of worry that this algorithm could not work at all. So, but that's good that it's working out.
Yeah, like first of all, I was really skeptical, like it might not work because in our case as well, we have to change a lot of things in the algorithm to make it work to basically generate English text into Japanese. But in this case, as Smith was also suggesting, like because the shape of the basically Basileia is very similar throughout, but the English characters are very different. The Japanese is like 10,000 or something character set, so it was very difficult there to basically generate these images. But here it is working very fine, I guess. It's working absolutely fine.
Well, that's good. Yeah. Yeah, that's good. Any other updates on that did you want to make, or?
Not from my side.
Tharun?
Yeah, so like after this model, I think we should try those edge detection models. Like you're suggesting some canny edge detection and watershed algorithms. So that we just look into this this week also after training on this new data set.
All right. So the edge detection models would be on the output of these algorithms, or?
Yeah, so after finding these bounding boxes or something, we can extract those coordinates of the boxes, and then we can just track the box across different images.
All right. Yeah, that sounds good. Yeah. Yeah, so you'd have this output that's refined, and then you would be able to do some further computer vision on it, and then you'd be able to find some, yeah, for the movement part.
Yeah, the output would make it easier for these edge detection algorithms because it would just be a black and white image.
Yeah.
So it would be much easier for them to find gauges.
All right. Well, that's good. Thank you for that update. So yeah, that sounds good. And so so for those of you watching on YouTube here, welcome to the meeting. And we were just talking about the update on the Basilaria, more Basilaria stuff. So we've done the digital Basilaria project, and we're doing a second data set where we're doing some algorithms, applying some different algorithms to those images. So thank you for that update, Tharun and Ujjwal. And I thought I'd go through some other things here that we have. Let's see, today we have a number of things to catch up on. Some people here are interested in, I think we were talking about Neuromatch, which is coming up. And I wanted to remind you that we have the abstract submissions document. So if you're doing, you're interested in doing something for Neuromatch conference, we have our ideas board. This is with both this group and the other group that I work with. So you'll see that they're labeled here in this column. And you don't have to pick something from DW, which is the DevoWorm group. You can pick something from any of these if it looks interesting to you. So we have the ones that we've done in previous conferences. We did some work on the artificial and biological neural networks. We have some stuff on complex networks. We have this unified theory of switching. Some of these are not like easy to, some of these aren't, the title isn't straightforward. So if you're interested, inquire for, find out what this thing is that's listed here. And then if you're interested, you can put your name here in this interested collaborators column, and or you can just contact us on Slack and say, what is this all about? So you'll be contacting me probably because I'm coordinating this, but so we have a number of different things. Now, not all of these are going to make it as submissions, but we want to create a submission, and we want to pick the strongest ideas. So we have a number of different ideas here, and then we have this document here, which is a submissions document. So when we come up with an abstract, we'll put it in this file, and then you can actually browse these abstracts to find out more about the thing that people are doing. So right now we have a couple of abstracts that are candidates. Looks like Jesse Parent has been populating this with things. And so if you have an idea, you can put it in this format where you have the title and the body of the abstract. Please don't overwrite anything that's in here. We have a number of different ideas, and so just just pick one of these title body tags here and just put the title here and the body here, and you should be fine. But mostly you're just going to be looking over some of these ideas and saying, I want to be a part of this. And you can leave comments if you want to improve one of the abstracts, if you think it needs something else that we're missing, or if you have questions, you can leave comments in this document. So, and then we won't be just be submitting the NeuroMatch with this system. We'll be finding other conferences to submit to and putting more ideas in here. I want to get a system where people can sort of put ideas out there, make an abstract for it, and then people browse and see if they find something that they like, and they can become a collaborator on it, and then maybe we can submit it somewhere. So we'll have an abstract all ready to go for some conference. We can submit it. So that's what I want to. That's my aim for this. And so for people in this meeting, I will put this in the chat if you want to take a look. And this applies to Ujjwal and Tharun as well. If you have an idea for something we might do with the work that you're doing right now, put it in there. Put it in as a item, and then maybe you can put an abstract in. We don't have to do that today, but we're going to do this for future submissions. So this is good. And then of course, if it's like a machine learning conference, like something where they want a paper instead of an abstract, then still having the abstract there I think guides us in terms of where we want to go with that. So we can put out a paper. We have an abstract. We can flush the paper out and get it together for a conference, I think, quicker than we would be able to do otherwise. So that's that part. The next part is the submissions, overall submissions document. So this is the thing we've been working on from last year. And so actually, I think the beginning of this year, actually. And so this is everything in 2021 that we've been doing. There are a lot of things we've kind of missed, a lot of things we've hit, a lot of things we've been rejected from. Actually, not that many things, because the AOIF conference, for example, was a rejection. But we have a number of things that are outstanding. So there are a number of papers that, if you're interested in diatom movement, or in genomic research, or cell segmentation, those are things that, we have these items like the quantitative comparison of Archaea, and this NeurIPS workshops. I think that's come and gone in terms of submissions. So I'm going to put, I don't think anyone submitted anything successfully to that. So NeurIPS is actually coming up at the beginning of December. And if you're interested in machine learning, deep learning, that's a good place to just attend if you can. It's going to be virtual again this year, so if you want to attend, I think student rates are lower than the regular rates, so if you want to attend, try to register early for that. They'll also have a lot of materials up on the web where they'll have different workshops and different sessions that are open to the public, sometimes after the fact. So maybe what we'll do towards that period is try to focus some of our efforts around promoting some of those materials. So as it turns out, NeuroMatch 4.0 and NeurIPS are happening right around the same time in early December. So maybe we'll try to do like a DevoWorm centric review of some of the content, some of the ideas that are going around at these conferences, and try to align ourselves a little bit with that during the conference. I was thinking of doing something with OpenWorm. I don't know if that's going to happen, but they're doing these watch parties in person. But I was thinking of doing something like sort of a virtual group, maybe during one of our meetings, where we kind of recap some of the things going on in these two different conference venues and seeing what kind of fits into what we're doing here. So there are a lot of, NeurIPS is extremely large and has a lot of ideas floating around, and it's easy to get lost in everything that's going on. in my other group, especially with the help of Jesse, we've been able to distill a lot of this stuff, and kind of focus on interesting ideas, interesting to our group discussions. So maybe we'll try to do something like that, and I think that would be useful for a lot of different people, like Ujjwal and Tharun, who are interested in new algorithms, but also people who are interested in some of the larger ideas. If you're interested in some of the I don't want to speak for you guys, but if you're interested in some of the broader ideas that they have at the conference, broader concepts, if it's interesting to you, then please, we should talk about it during that time. I think that's a good opportunity to sort of decompress a lot of what's happening at these conferences, because there's way too much going on for one person just to kind of, try to distill on their own. And so that's what's going on there. And of course, Neuromatch 4 here, the abstract submission is October 25th. So that's coming up soon. So once we get those out, then, those will be accepted as either flash talks or 15-minute talks. And then we can start working on those presentations. With NeurIPS, we haven't submitted anything, but the conference is coming up, and it'll be available, so we'll be able to see what the state of the art is in the field, and so we can get an appreciation for that and maybe apply some of that to the work that we're doing. So I don't think there are any other pressing deadlines. I think that, let me put a red mark on this for Neuromatch. So this red mark means it's coming soon. Actually, I don't know. Oh, okay, that's workshops. So the red mark means it's coming soon. The red, if it goes across, means we're not doing it or it was rejected or something. Actually, I had a different color scheme here. It was grayed out. The text was grayed out, so maybe I'll just do that. All right. Oh, now let's do that. Let me do this. All right. There we go. I think I made a distinction there. Good. All right, so that's good. We have those two things coming up, and some of these things are kind of open, like this Williamson symbiosis, the diatom motion jerkiness, and so forth. So those are all open to collaborators, and I think a lot of the stuff we're doing with diatoms here in this Tharun and Ujjwal and Asmit, those are all sort of fit into one of these diatom categories. So we'll talk more about how we're going to submit that work later. So I think this is Krishna. Welcome, Krishna.
Hello. Has the daylight saving time started?
No, it has not.
Okay. So we are doing it 1 hour before.
No, I had to move it for this week because I had something else. But
okay. I thought like the daytime saving time has started, so that's why.
Oh, actually, it starts in a couple weeks, so that'll be a shift more generally. So yeah, I always make a mistake when on the onset of daylight savings time because I'm trying to coordinate different time zones, and I think in the
it really gets confusing.
Thanks for attending, Ujjwal. Yeah. So yeah. Okay. Yeah. Great work, Tharun. That was good. Yeah, so that's good. And so Krishna, did you have anything to update us on?
No, I was working on the abstract of the Neuromatch, but it's not completed, and the topic I've chosen is relatively new to me, so I'm currently now reading research paper. So I guess within 1 or 2 weeks the abstract would be completed. If anyone from this group, like Tharun, if you are interested in Neuromatch, feel free to collaborate. So have you shared the list of all the proposed topics in this group, Bradley?
Yeah, I think I put a link in there.
Yeah, it's in the chat.
Yeah, I may have to re-up it because I don't know if he'll see it. But yeah, this is the document here. And so that was, I just covered that actually, so it's on the recording, but just kind of going through that document, and we list like different things we want to submit. Then there's a link to the abstracts, or maybe we put an abstract in there, and then we can make a determination of how we want to. If we want to submit it to Neuromatch, or maybe we submit it somewhere else too.
So neural decoding is a relatively very new thing for me, and I'm still figuring out things. So let's see where it goes.
Yeah, that was the one that you had proposed in your neural decoding.
Yeah.
Yeah.
So how you have any experience you have worked earlier on neural decoding?
Not really. No, I don't. I mean, I know a little bit about it, but I'm not. Yeah.
Yeah.
It's that one. Well, that's the one where they do. I think they are able to extract movies out of fMRI data or something like that. Yeah.
Something like that.
It's interesting, but I mean, it takes a lot of algorithmic overhead. Like, I don't know what how they were doing that exactly, but.
So Susan hasn't joined.
Well, it's Canadian Thanksgiving today, so she's out. And so holiday for her.
Okay.
Yeah. All right. Well, let's see. I can go back and I want to continue on with some of the stuff. So we have this abstract submissions down here. Next thing I wanted to mention was that there's this conference that starts today and it goes on, I think, through this week. And it's called Combine. And this is a conference on computational modeling in biology. So this is something that we do here a lot, but I hadn't heard about this until last week. So I'm going to attend a couple of sessions here. This is focused around systems biology, coordinating various community standards and formats. So this is all on Zoom. And the reason I bring it up is because Stephen Larson from OpenWorm will be talking about 10 years of OpenWorm. Let me see if I can find I don't know if we have the agenda here, but he's going to be talking about 10 years of OpenWorm this afternoon, at least my time. It might be too late for Tharun and Krishna, but I don't think they have the event. I don't think they have the schedule up here. It's hard to get to because they have it in a special portal. But anyways, this is the event. Stephen Larson's going to talk about 10 years of OpenWorm. I think this is all going to be on YouTube, hopefully. So if you don't catch it, you can catch it later. But this is something we talked about, I think, last week, where OpenWorm is 10 years old. They've been doing this since 2011, and there's a lot to kind of catch up on in terms of what's the progress made, and what's the future look like. I think there's been a lot of talk about progress made because there's been a lot of progress made, and some people are more of the mind that the C. elegans brain should have been quote unquote simulated by now. But of course, simulating a brain is a little bit different than kind of what we've been doing, which is kind of approximating the brain and approximating the biology of the worm. So this is going to be an interesting set of sessions. I think Steve Larson's talk will be really interesting. There are a lot of interesting talks in here in general. So there's a lot of stuff on visualization, on data, open data pipelines and model standards, and standardization of AI approaches in biological modeling, multicellular modeling, and all these other things. So I think this will be a nice event. I don't know if you can still register. Let me see if I can give you a link. I don't know. It's kind of opaque as to how to register for this. So I don't know. Maybe you can find it through this web page. Yeah. So there's that. And hopefully we'll find out more by next week. Let you know how the talk went and everything. So last week I talked about the Nobel Prize actually last week was something I called Oct4 Day. And the reason I called it Oct4 Day was because there's a technique called cellular reprogramming where they take some sort of somatic cell and they can turn it into a stem cell. And to do that, they introduce what they call they overexpress something called a transcription factor in the cell. So this is something that is created by the expression of a gene, and it's an mRNA molecule, and they get expressed at a certain concentration, and they affect different other the expression of other genes. They activate different pathways, and you end up ultimately, if you have the right mRNAs expressed at a high level, it can affect the physiology of the cell. And in this case, you have the people won the Nobel Prize that identified certain factors in cells that make them change what they call transform from one phenotype to another. So, they identified, I think, 4 or 5 transcription factors that were responsible for creating a stem cell from a somatic cell. And one of those transcription factors was called OCT4. So we talked about that Nobel Prize. That was in 2012. That was Yamanaka and John Grudon who did a lot of work on reprogramming germ cells. So taking a mature differentiated cells nucleus and putting it into an egg cell, and then that would create a clone, clonal organism, and the gene expression program in that case would be rewritten for that cell, and it would behave as if it were like a germ cell from a different organism. Cool stuff. So that was the 2012 Nobel Prize, and surrounding that. And I mentioned something about the 2021 Nobel Prize for Physiology and Medicine, and that is something actually quite different in terms of what the award was for. But it was just released last week, right before the meeting, so I didn't have a chance to actually read through some of the material on it. So I promised that I would present it today. And so with that long-winded introduction, I wanted to talk a little bit about this, because it's actually interesting in a neuroscience sense. So this is another, this prize is very neuroscience-centric. So this is the 2021 Medicine Nobel Prize winner, explains the importance of sensing touch. Artem Pataputian, who shared the Physiology or Medicine Prize for work on mechanisms crucial to everything from bladder control to knowing where our limbs are. And so this prize was about cells that sense touch or pressure from the environment. Pressure, heat, that sort of thing. So one of the discoveries surrounding this prize was the discovery of what they call capsaicin. And so if you've ever eaten hot peppers, there's this chemical compound called capsaicin. And it hits your tongue, and it burns your tongue, and you have to get water to cool it off. That burning sensation is actually a touch receptor or a pain receptor that's activated in the tongue when you eat that, when you're exposed to that compound, and it triggers a response in your brain, in your nervous system, and it's transduced from the environment into the cells and then down the nerves. And so as it turns out, there are a number of different types of cell like this. Of course, if you feel pain, you have pain receptors that do this sort of transduction. You have proprioceptors that do this. So if you're moving through the environment, you feel your body moving. You can, you're aware of like where your arms are, your legs are. Those are all proprioceptors that take in tiny amounts of force from the environment, and they kind of, can tell you kind of where you are in space. And it's very subconscious. You don't really know that you're doing it. So the Nobel Prize here not only was about these receptors and knowing what they are, because people knew what they were. They've known, about these receptors for a while, but about 20 years ago, they started to find genetic correlates to some of these processes. So when you eat a pepper, say, and you bite into a pepper, and that capsaicin hits your tongue, it actually triggers a molecular cascade in that cell, and that actually is regulated by different genes that are regulated or that are expressed in those cells. And so let's see. So for example, in pressure-sensitive ion channels, known as piezo1 and piezo2, these are protein molecules that are involved in sensing pressure, and these are pressure-sensitive ion channels. So that means that when you have when you have this stimulus on the cell, the stimulus activates these pressure sensitive ion channels. When there's a certain amount of pressure applied to these receptors, for example, these ion channels are activated, and these proteins are responsible. So they're embedded in the membrane of the cells, and they either open or close these ion channels, and so they activate the cell. So basically, you're getting some environmental transduction. It's activating some molecule. It could be piezo 1, piezo 2. It could be, there's another one for the capsaicin example. They activate these ion channels, and then they activate the cell. And so then the cell is active, and then it transduces information from the cell and down into the nervous system where you feel it. And so now your brain is getting these signals, and they're regulated by these proteins at the receptor. And then all we need to know now is how your brain makes sense of them. That's a little bit harder problem to solve. So there was a lot of work that went into this. They had to figure out which genes were responsible for creating these proteins. They had to do a lot of what they call screens, which means they had to test a bunch of different genes in parallel and try to figure out the one that had the biggest effect on the thing that they were testing. So that's a lot of hard work. And so I think this is leading us closer to understanding what we call proprioception, and also pain reception and other things. So this is where there's a lot of unknown things here. It seems like we would know a lot about it, but we don't. So the genetic work on this is about 20 years old, and so this is interesting. This article is an interview with this scientist, and they talked a little bit about why this sensing temperature is important in touch. So it's temperature and touch kind of combined, but this is one mode of sensation that we don't really think about too much, but it does play a role in our cognition and in our behavior. So it's fascinating how one of the 5 major senses mechanistically, how we sense touch, was not really understood. It's something so unique because everything else, whether you're talking about smell or taste, which are based on chemicals or hormones being secreted from your body, such as insulin, is chemical sensing. So this is a phenomenon that's completely different, and it's based on sensing physical stimuli such as pressure. But I also find super fascinating this idea of proprioception, the sense of where your limbs are compared to the rest of your body. I consider it perhaps your most important sense. I would say a majority of people have probably never even heard of it or never stopped to think about this sense. And so your sensory neurons actually innervate most of your body, your muscles, your skin, everything. And so this is the mode of sensation where your muscles and your skin are all kind of monitored and detects changes in your muscle contraction, changes in your skin. If there's you put a wet towel on your arm, you're able to sense it without having your eyes open. You can do this, and it's an interesting set of experiments if you decouple vision from proprioception, but that's another conversation. And so this is all very interesting, and it's not very well known as to how this works. We actually know, in fact, that C. elegans, this is an important mode of sensation in C. elegans, because there's a lot of sort of touch reception in C. elegans, and it especially affects the way that C. elegans moves around its environment. C. elegans will touch you can do these experiments where you sort of brush a C. elegans on the nose or on the anterior end with what they call a worm pick, and the worm will sense that and detect it at a very fine sort of grained level, and then move away from it. So it knows exactly where you're putting that pick. It's not smelling it with using chemosensation, and it's not seeing it. They don't really have eyes, so they use this sensation. Yes, Krishna, would you like to say something?
It's like a little bit related to the topic. Have you heard some people who don't have vision, they are blind, they have loss of vision, they have very good sensing, like their skin can sense better than normal people. So, is there any correlation between one sense and the other? Because I've heard like some scientists or artists, they usually touch different things or they listen to music. So they are using their senses in one way, and it simulates their senses in the other way. Like if you want to uh, how do I say it? If you want to compose, I don't know, it's true, but it said like if Beethoven has to compose a music, he would see art pieces or paintings, and that would inspire. And some of the blind people, they have very good sense, like this. How do I say they have good sensory abilities in touch and this. So, is there any correlation between different senses?
Yeah, so there is a whole field of what they call sensory integration or cross-modal plasticity.
Yeah.
And so you can, like in humans, and not so much in C. elegans, although C. elegans has some of this, where you have two different senses that are taking in information from, kind of being in the same place. So if for example, in a
If we
human,
give them simulation, yeah, is there literature on this thing? Like, if we give them one simulation, does their other sensory organs would be more? You can say, how do I put it? Like, they will be more exaggerated, something like that. If you put them into like high luminous intensity, is there chances that their sensory would be affected by that? Any
yeah, so
literature on that?
yeah, so what happens, I think, in people who go blind early, they found that in humans, because in humans we have a large neocortex that has devoted to processing different senses. If you were to take away
Yes.
vision very early, you don't have the entire visual cortex is not connected to the visual system. But what happens is that the other senses recruit parts of that cortex, and so they have this heightened representation in that part of the brain. So the touch areas or the touch sensation and the auditory sensation, they recruit those areas, and so they have a larger sort of representational canvas, and that takes the place of vision in those people. So yeah, there is a lot of that sort of cross modal plasticity. I can guide. I guess if you just look in under cross modal plasticity, you can find some references, but I have some resources as well on that. So
So, thank you.
you're welcome.
Yeah.
So yeah, I wanted to talk about. All right. That if you have questions about, it, we can talk about that later. I did want to go on to this other thing that I found. I was reading about this blog post here. So this is Jonathan Gordon. This is about something called deep sets, and it's a general introduction to deep sets and neural processes. So this is something. I think this is. I don't know what this is based on. It's just kind of a blog post. For right now, we'll just consider this separately. So this is a diagram of sort of the deep sets that they're talking about here. So in this post, I'll discuss two topics I've been thinking about a lot recently: deep sets and neural processes. And so this is about meta learning, actually. So meta learning is this learning from learning sort of idea. So in the standardized supervised learning setting, we are often interested in learning or approximating a function that maps inputs to outputs. So this is kind of like what Pix2Pix does. A supervised learning algorithm may be considered an algorithm that, given a data set of such input-output pairs, returns a functional approximator. If L is a good algorithm, F is equivalent to F, F hat is equivalent to F in some meaningful sense. So the function approximator is equivalent to this function that maps inputs to outputs. So the idea is like, you're trying to approximate that mapping and you want to get close to it. And that's what you're doing here. In the meta-learning setting, rather than assuming we have access to one such data set, we assume that our data set is comprised of many tasks, each containing a context set and a target set. So this is kind of like the mapping between these two. Each of these in turn contain a variable number of input-output pairs. So now instead of having just one mapping, you have a variable number of them. Our assumption is that while the mapping between inputs and outputs may differ across tasks, the tasks share some statistical properties that, when modeled appropriately, should improve the overall performance of the learning algorithms. So the goal of meta-learning is to produce a black box algorithm that maps from data sets to function approximators. Our goal is to use data of tasks to train a model that accepts new training sets and produce function approximators that perform well on data that's not seen before or is not labeled. Meta-learning learns a learning algorithm that is appropriate for all the observed tasks. So that's learning to learn is. That's what meta-learning is. So one of the most compelling motivations for meta-learning is data efficiency. So your average setting requires a lot of data to learn. So we want to reduce that amount of data. We want to make the learner more efficient on samples that are presented to it. If we can do that, then we can, this is something we can do with something like few-shot learning, where we only introduce a couple of examples and it'll pick up the larger category. That's basically what we're trying to do. So here's some examples. This Segway scooter, does this generalize to different types of wheeled transport? Or does this letter approximate a number of other letters in a writing system? So that's something that you want to try to test given your algorithm. And if you can get few-shot learning, you can maybe introduce a couple of different types of wheeled transport and get the entire set of wheeled transports that exist. And so that's the kind of thing you're trying to do with few-shot learning. Now they talk about neural process family. So the neural process family is a recently introduced family of models for probabilistic meta-learning. So in this post, it'll focus on conditional neural processes, which are a simple deterministic variant of neural processes. So this leverages a series of mappings that directly model the predictive distribution at some location of interest, which is x sub t, conditioned on a context set. So this is the mathematical definition of it. So y sub t is the input we wish to make a prediction at. D sub c is the context set to condition on. And so this is the computational structure of a neural process. This is an encoder-decoder perspective here. So we can build this. This is an encoder and then the decoder from A. And so on the left, we have these context sets, X and Y, and then that represents the context set. The E block is the encoder, and then the alpha, or the A circle, the pooling operation. So this is the pooling operation. These are the encoders here, these E's. This is the pooler. And then D block is the decoder here, which outputs to these different types of distributions. So this is the way it's basically set up. So this kind of goes through the math, and then this shows the training procedure here. And then deep sets are kind of an extension of this. It goes into the inner workings of this neural process here. A useful way to think about them as follows. Our decoder is a standard neural network that maps X to set Y with one small tweak. We condition it on an additional input R, which is a dataset specific representation. So this is an input that tells you something about the dataset. The encoder's job is really just to embed datasets into an appropriate vector space. CMP specifies such an embedding and provide a way to train this embedding end-to-end with a predictive model. So is this the correct form for such an embedding? So what does a function that embeds a data set in a vector space look like? So this is different from a machine learning model where we expect inputs to be an instance of some space, like a vector space, or maybe sequences. What we want is a function approximator that accepts input sets. So we want to have a set of inputs or treat it as a set instead of as a space. So what are the properties of sets, and what properties do we want such a function to have? So sets have varying sizes, and we want to be able to handle these arbitrarily sized sets. So, you have different types of things in the world they don't all have the same number of samples in them. Sometimes they're very small, sometimes they're very large, but you want to treat them sort of and normalize them across sets. The other thing is that sets generally have no order, at least in this context. Your sets are just kind of there. There's no real information about ordering. It's just kind of like information about this thing, this category. And so any module operating on sets must be invariant to the order of the elements. This is known as permutation invariance. And then so this makes for a hard problem because some of these things aren't really amenable to quick and easy learning, but that's exactly what we want to do. So they give some examples from before. They give some previous examples from earlier research on this problem, kind of going through some of these things. And then the summary is that we've taken a first look at neural process families, these model families that allow us to do probabilistic meta-learning. The modeling of sets is also a very interesting subfield. It's at the frontier of machine learning research, but it's not really very well developed. And then, so this is, yeah, I'll be talking more about some of this stuff later in other posts. But this is something that, if you're interested, maybe we follow up on this. I'm not really sure how hard this is to implement or if this is related to some of your work, but this post is definitely maybe will be useful to some of you, especially in terms of thinking of inputs as sets. We haven't really talked too much about like some of these things, but I think we talked about category theory in my other group, and this is very similar in some ways to category theory, although the mathematics is very different, and the way you treat the input sets. They don't use sets, they use categories, they use things like functors and other terminology, which make this problem a little bit different. But it's very similar in spirit in a lot of ways to what's going on here, where you don't have labeled data, you have sets of instances and things that are in, not necessarily in ordered categories, but they're sort of semi-structured. And so you're putting them into this model and you're trying to learn from some of the rules that already exist. You're trying to do this meta-learning, and so this is an open area. It's very I don't think there's a lot of hard and good results to show. It's just kind of what people are thinking about how to make this, how to do meta-learning and how to do this better. So okay, I was going to mention that this is again Hacktoberfest month, so this is the Digital Basilaria repository, and we have the badge here on this repository. We also have the Hacktoberfest badge on the group meetings repository. And so if you look here, this is more along the lines of different projects that are ongoing. So if you go to this group meetings project board, you can see some of the things we're involved in. If you want to participate in some of these things, pick an issue and mention it in the Slack. Say I'm interested in issue 90 or 100. Actually, 100 is finished, but some of these issues are live and they're very interesting. So if you want to pursue some of these things, ask about it by number and we can discuss it. And maybe, you can make a contribution here. We also have the Hacktoberfest badge on DevoLearn, which is, of course, the software and learning platform that we have for DevoLearn for, machine learning. We also have data science demos that a lot of people have contributed. So if you're interested in contributing a short demo on some data science topic, you can do that here. You can contribute to the DevoLearn software, which is somewhere in here. It's right here. Or you can submit to some other repository in this organization. So that's the Hacktoberfest is going on now through the end of the month. So finally, I wanted to finish with some papers that we've been talking, or that maybe are related to some of the things we've been talking about. And I'm going to go over the top of the hour a little bit to do this. One of these is this paper on multi-layer C. elegans connectomes. So this is a newer paper. This is by Avyamini. He's worked with the OpenWorm Foundation before. Ed Bullimore, who's a person who does a lot of fMRI work, especially networks, and William Schaefer, who's also been involved in OpenWorm quite a bit. So this is the multi-layer connectome of C. elegans. So the abstract reads, connectomics is focused primarily on the mapping of synaptic links in the brain. Yet it is well established that extrasynaptic volume transmission, especially via monoamines and neuropeptides, so these are neurochemicals in the brain, is also critical to brain function and occurs primarily outside the synaptic connectome. We have mapped the putative monoamine connections, as well as a subset of neuropeptide connections in C. elegans, based on a new and published gene expression data set. So they're basically looking at the expression of neuropeptides, the expression of monoamines, and so this is something that occurs not like within the existing synaptic connections, but actually as signaling molecules. So our embryo networks that we talk about in this group actually are based on the principle of, paracrine signaling, or cells that are near each other sharing chemical signals with each other. And so this is actually something that is in that direction, where we can map this out. We need to understand how genes are expressed and what they're producing in terms of chemicals that allow for this type of thing, this type of data set to be constructed. So these type of networks exhibit a distinct topological set of properties. The monoamine network displays a highly disassociative star-like structure with a rich club of interconnected broadcasting hubs. So these are cells that are putting out the signal into the extracellular matrix. And in fact, this is a broadcast model because it's broadcasting from the cell outward. And so, you usually have this area of influence where the signal is great and then it decays sort of linearly as you move away from the cell. So the idea would be that you have certain cells that broadcast a lot of this, and they're rich because they reach a lot of other cells. And the neuropeptide network showing a more recurrent, highly clustered topology. So the neuropeptide network is being expressed at maybe shorter ranges. Maybe it's being expressed more regularly across cells and the like. So there are differences in these two different types of chemicals and how the genes are being expressed to produce them and their diffusion. Despite the low degree of overlap between the extrasynaptic or wireless and synaptic or wired connectomes, we find highly significant multi-link motifs of interaction, pinpointing locations in the network where aminergic and neuropeptide signaling modulate synaptic activity. So these chemical signals that are out there, they actually modulate synaptic activity, which are also outside of the cells. They're in in the space in between the cells. So they're also regulated by this. So synapses are these connections between two cells. Usually the cell sends out an axon, and there's some junction between cell processes that meet, and this is where a lot of the chemical communication occurs. These chemicals that are sent out into the extracellular area will affect this sort of transmission milieu by interfering or augmenting what's being transmitted at the synapse. So it's usually things that are being exchanged across this short gap between the cell's processes. So that's to give you an idea of what that looks like. This C. elegans connectome can be mapped as a multiplex network, meaning that it has multiple layers. One of the layers being synaptic, another being this chemical a map that they're using, and neuromodulatory layers representing alternative modes of interaction between neurons. This provides a new topological plan for understanding how these types of chemicals modulate behavior and how this is achieved by specific motifs and loci of integration. So this is let's see if they have any images here for this. Now they talk about these networks, and I think we've seen the C. elegans connectome, but this is kind of an idea of what we're dealing with here. So these are the receptors, expression patterns of dopamine receptors. So these are in the C. elegans, sort of images of a C. elegans worm, an adult worm. And these images here show some of these receptors, very high density. And so the idea is that these receptors will take up these chemicals and use them for different processes. So, some cells have large areas of receptor, so they're able to visualize these in the microscopy here. And they're able to identify which cells are sensitive to these different compounds. So this is actually their, let's see. This A is, these are the RAM tyramine releasing neurons in A. So you can see that there's this core of neurons that are, where there's a release from these two neurons that are affecting these cells. And then in B, we have RIC, octopamine releasing neurons showing outgoing synaptic edges and neurons expressing one or more of the three octopamine receptors, gray. So this is, again, where you have a subset of neurons that are putting out this chemical signal, a subset of neurons that are taking it up, and then the rest of the neurons here around the edge. So this is how they construct this connectome. And then D is this multi-layer expansion of the synaptic gap junction, monoamine, and neuropeptide signaling networks. So you can see that the topology changes as you consider different types of, so you have a gap junction network, which is where you have cells that are adjacent and they share what they call an electrical synapse, which is a gap junction. They have other types of, you can use different criterion to build different types of network. That's the key takeaway from this. And so it looks very different depending on what you're considering as the basis for that network. And so then they have a table showing the number of receptor expressing cells that do not receive synapses from releasing cells, and the number of connections in each layer that are non-synaptic, including connections between neurons within the same class. So this is cells with no synaptic input. These are non-synaptic edges. So these are for different neurotransmitters, and you can see that there are differences in the numbers. And then this forms a multiplex network. So the full C. elegans connectome can be considered as a multiplex or multi-layered network. You can put these different network topologies on top of one another because they sort of function, the entire nervous system has all these different things going on simultaneously. So you can stack these on top of one another and say that this, when you're considering one type of neurotransmitter versus another type of neurotransmitter, the topology shifts. And so then you can say things about behavioral circuits. So if you consider a behavioral circuit, some behavioral circuits are reliant on some types of neurotransmitters, but not others. So this is different from another type of behavioral circuit. Say, like, for example, the hunger circuit, the circuit that controls satiety of finding food versus one that's involved in navigation. And so those circuits are going to be different, not only in their structure, just in their gap junction network structure, but in the way they utilize neurotransmitters. Let's see if there's any other nice stuff in here. This is the monoamine rich club. So this is a rich club is where you have maybe a couple neurons that are doing most of the work in the network, or that's connected to most of the other cells in the network. And this is not really that understandable, this graph. This is a graph that they like to use for visualize what they call hive plots. So they're showing like the connections between different types of functions, so sensory, motor, and interneurons, how they're connected together using these criterions. So your interneurons, your sensory neurons, and your motor neurons all have different types of connectivity here. You can see if you break it out in those categories. So that's all I'll talk about in that paper. And this actually shows neuropeptide networks across different types of neuropeptide. This is really cool. It has all the different molecules that they've identified in this study, and their different topologies. So you can see some are very small networks, some are like this fan-like thing where they have one very highly connected node here, and then the rest are kind of like peripheral. You have others that are other networks that are more densely connected. So these connections, these have meaning. You can characterize these using a statistical parameter, but you can also, it does have meaning in terms of, the networks, the cells that are key in the network. So if you were to knock that cell out, the network would collapse. And so if you imagine if you knocked out a cell, it would collapse maybe for that neurotransmitter function, but the other neurotransmitter functions might retain their ability to connect to other cells. So, and this is something you might find if there's like some mutant that has, some cell is deleted or some neurotransmitter is deleted, that will affect the behavior, and you can trace it back to these types of networks. So that's all I'm going to talk about for that today. I'm going to talk about one more paper, and I think this one here, this would be good here. So a couple weeks ago we talked about, I think it was Akshay who's working on tiling the sphere for the axolotl atlas. So he's taking the axolotl data and he created a sphere in, I think it was in Python, where you have this three-dimensional sphere that you can move around and explore, and then you can you tile it with these microscopy images of the axolotl embryo. And so we have the data to create this map. And so you have this thing that's on a sphere, right? You'll have this model that's on a sphere, and so now you have this embryo. But it's not dynamic. It's just kind of the static image. It could be dynamic if we put different types, images from different time points on it. But then the question is like, okay, we have this changing set of images on the sphere. Now, what about representing dynamical systems on such a sphere? If we can make like a sphere, a static sphere of the axolotl embryo, and then we can extend that out to multiple time points, then can we go a step further than that and create dynamical systems on the sphere? And this is what this paper is about. It's about creating a Kuramoto model on a sphere. So a Kuramoto model is this. This is Steve Strogatz, who's a big name in complexity theory, Renato Mirolo, and Max Lipton. This was in Chaos Journal. So Kuramoto models are these models of synchronized oscillators. And so you might have an oscillator for like neural activity. You might have an oscillator like something like a firefly that's flashing. It's, they generate light through a sac of luciferase that they have on their back end of their body, and they flash it as a means of communication. And this flashing can occur at different frequencies. So this is an oscillator that oscillates at a certain period, or maybe it's unsynchronized. In groups, they actually synchronize their behavior. And Kuramoto was a scientist who created a model of this type of behavior, this sort of synchronization of these oscillators. So you can apply it to, fireflies, you can apply it to neurons, groups of neurons. You can apply it to anything that has an oscillation period. If you have a lot of agents with different oscillation periods to watch them synchronize. So this is the title here. We study a system of n-identical interacting particles moving on the unit sphere in d-dimensional space. So it's just this bunch of particles on the surface of a sphere, and they're interacting with one another. The particles are self-propelled and coupled all-to-all, which means there are no, pairwise dependencies are just kind of like associating with every other particle. For d equals 2, the system reduces to the classic Kuramoto model of coupled oscillators. For d equals 3, it has been proposed to describe the orientation dynamics of swarms of drones or other entities moving about in three-dimensional space. Here we use group theory to explain recent discoveries of that. The model shows low-dimensional dynamics for all n greater than 3, at or greater than 3, and to clarify what it admits, why it admits the analog of the antons and ansats in the continuum limit. So I'm not really sure about what that means. It's beyond my advanced skill set in chaos theory, but The underlying reason is that the system is intimately connected to the natural hyperbolic geometry of the unit ball. In this geometry, isometries form a Lie group consisting of higher dimensional generalizations of the Mobius transformations used in complex analysis. Once these connections are realized, the reduced dynamics and the generalized form of this follow immediately. This framework also reveals a seamless connection between the finite and infinite n cases. So finally, we show the special forms of coupling yield gradient dynamics with respect to the hyperbolic metric and obtain globally stable results about convergence of the synchronized state. So I think what they're trying to do here is they're taking this model, they're taking these, I'm just kind of hoping for some visualizations here, but maybe we'll get them. I'm not sure. But they're taking the sphere and they're looking at things on the surface of the sphere, and they're able to look at different densities. And then they're able to look, okay, so this is a first order linear Kuramoto system. So you have these different states where you have different sort of how they migrate across the thing. And the idea is they'll either cluster like this or they'll be all over the place like this. And so you can see that they're, either they're scattered and random or they're synchronizing in one location. And this was generated in Python, visualized with Plotly. So you can do this sort of a simulation where you have this surface of a sphere with all these particles, and they're, you can see if, you can use this type of system to observe, with different densities of particles to observe synchronization of behavior. And so I don't know if you need to apply really high-level math to get some interesting results in something like this. The reason I show it in the group is because I wanted to show what this looks like. I wanted to show that, with this sort of spherical model, you can obtain a lot of different interesting things, different types of interesting results. So in this case, the first order linear Kuramoto system with weights distributed according to a Riemann sum approximates the integral of a normal distribution of randomly chosen initial conditions. These pink particles here contribute to the order parameter with greater weights than the blue particles do. So you can see that there are these pink particles are contributing to this to order more than the blue particles, and you see that the pink particles disappear here. These are for different initial conditions. So you have different initial conditions to start off with, and they result in this sort of distribution of particles. And so in this case, it's a different type of criterion, so you see different results here. And that's all there is to that paper. So, I mean, this is a bit beyond what we're kind of... Okay, so thanks, Krishna, for attending. So anyways, that's all I have for today. Next week, we'll talk about... I think Susan might present on her materials that she's been working on. She's been working on some physical models of the embryo and some signal processing stuff. I don't really know what she's going to present on. Also some soft materials. It's going to be a mix of things that she's going to focus on. I don't know if she's going to present next week, but she said that she might be able to do it. And so I look forward to that. And if you have any updates on the Basilaria work, if we have Akshay, maybe he can update us on the stuff he's doing with the axolotl embryos, and anyone else, if they want to present something, they're welcome to do it. So thank you for attending, and talk to you next week.